PCR go? Well, you need the usual PCR reagents: DNA, primers, dNTPs, DNA polymerase, and thermal cycling. But you also need an extra ingredient. One example of that extra ingredient is the intercalating dye cybergreen. This is how cybergreen works. In free solution, it does not glow. In the presence of double-stranded DNA, however, the cybergreen will intercalate in the DNA and glow green. Cybergreen does not interact with single-stranded DNA, such as the DNA in the oligonucleotide primers. So in the presence of the primers, it will not glow green. It will only glow green in the presence of double-stranded DNA. So this is how the cybergreen strategy works. Look up here, we've got a solution of cybergreen. And as we amplify our target DNA, the cybergreen has an opportunity to intercalate and glow green. And as we get more DNA, the cybergreen has more opportunities to intercalate and glow green. So the cybergreen is an indicator for how much double-stranded DNA is being amplified. Now, there's another strategy that uses a reporter oligonucleotide. And this reporter oligonucleotide is complementary to the target DNA and will hybridize, but it's specially constructed so that one nucleotide on the reporter contains a quencher, and another nucleotide on the reporter contains a fluorescent dye. Because the fluorescent dye is close to the quencher, it fails to glow. But on release from the oligonucleotide, it is no longer close to the quencher, and it can glow red. So here's how the reporter oligonucleotide works. Suppose we have the target DNA, and we have a primer oligonucleotide. In addition to that, suppose the reporter oligonucleotide also binds to the target DNA. DNA polymerase comes along and bumps into the reporter oligonucleotide. The DNA polymerase that's used in this reaction, in addition to having a 5' to 3' polymerase activity, also has a 5' to 3' exonuclease activity. So as a result, the polymerase can chew through the reporter oligonucleotide, releasing the reporter oligonucleotide first, releasing the oligonucleotide bound to the fluorescent dye, but now that's free and no longer near the quencher. And then it releases the other single nucleotides until it reaches the quencher, which is no longer close to the fluorescent dye and therefore unable to quench it. And as you go through multiple cycles of PCR, there are more and more opportunities for the reporter oligonucleotide to hybridize to the target DNA, and more opportunities for it to release its fluorescent dye. So either of these strategies is capable of reporting on the amount of DNA in any one of several reactions. So you could search around for, say, a plate with lots of wells, so you can do a lot of experiments in parallel. And look, down here. Here is a 96 well plate. Well, I'm going to use it and put a whole bunch of reactions into the 96 well plate. And each reaction is somewhat different. And I expect each reaction will have different amounts of the target DNA. So if I were using cybergreen, for example, Some of the samples will glow a bright green, and other samples will glow less brightly, indicating different concentrations of the target DNA in each well. And there's a way of doing this that allows you to be very quantitative. Here's an example of qPCR data for 5 different samples. Each sample was run in triplicate. So for example, the green sample had the highest concentration because it reached the threshold signal in the fewest number of cycles. And the turquoise sample over here had the lowest concentration because it required the greatest number of cycles to reach the threshold. So, in summary, there were 2 strategies for quantitative PCR. One was the cyber green strategy, and that's cheap because all you need is a fluorescent dye with the right properties. The second was the reporter oligonucleotide strategy. And that's much more expensive because you have to chemically synthesize an oligonucleotide tailor-made for the target sequence you're interested in, and therefore it costs more. However, the reporter oligonucleotide is more specific. Because a signal is produced only if the reporter oligonucleotide hybridizes to the target DNA sequence. So you have the specificity of the 2 oligonucleotide primers plus the reporter oligonucleotide. So there you have it, quantitative PCR.